such as transpiration also leads to regulation of stomata conductance. And sorry, it is regulated, or leaf temperature is regulated through stomata conductance and also modulated by the humidity and also the wind speed. And other factors such as the airflow around the leaves and the canopy also influence the leaf to air temperature difference, which also affects the energy balance. Now, things that determine the leaf temperature includes both, as I was saying earlier, both biotic and also abiotic factors. And with the abiotic factors includes factors like air temperature, the strength of the solar radiation, and also humidity, which affects the vapor pressure deficit. And moving on to the biotic factors, leaf traits such as the stomata conductance and then the leaf size and then shape, which also affect the leaf boundary area. And then the angle of the leaf, which also affect the solar radiation, which is heating the leaves. And then also optical properties such as reflectance and then absorbance also leads to differences in leaf or canopy temperature in general. Now, bringing into context the unique phase experiments that I am happy to be part of, the effects of elevated CO2 also has an impact on the leaf temperature, in that studies have shown that stomata conductance is reduced by a percentage about 20%, according to Edwards and then Rogers, and that has implication on the leaf's ability to transpire, and also through transpiration, the leaf temperature is also regulated. So in this case, if carbon dioxide is increased, then that means that leaves are going to get hotter because the stomata conductance is reduced. They will not be able to transpire enough water to cool down the leaf, and it will be worth or interesting or important to look into how leaf or canopy temperature is also faring under elevated CO2 conditions. Now, the reason why we need to also consider leaf temperatures in the various models that we are undertaking, climate models that we are taking, is that it affects photosynthesis. And because leaf photosynthesis are net carbon isolation increases to an optimal level, and then it begins to decline. Though even though it may be able to acclimate under higher temperatures, instantaneous increase in temperature can affect the rate at which carbon is being captured. And also it affects respiration as well. And then lastly, it also affects the photosynthetic thermal tolerance of leaves, because one of the most sensitive or temperature sensitive component of the leaf is the photosystems, which is affected by high temperature, in that beyond a certain temperature threshold, its ability or its efficiency to regulate photosynthesis also begins to decrease. So it is worth understanding or studying the effect that increased temperature can have on leaves. Now, for 2022, my main aim was to look at what temperatures are reached by the mature leaf of oaks under in heat wave conditions, and also whether the increase or the influence of the elevated carbon dioxide affects the temperature that is being experienced by the leaves. And then going further into the leaf tolerance, also see the upper temperature tolerance of cocos rubor leaves, and whether the increase in carbon dioxide is affecting the thermal tolerance of the leaves that are in the site. And then to see whether that tolerance or the upper temperature tolerance is affected by heat wave exposure. So tying this to the theme, the trees in time, my work focus or the time frame or temporal frame ranges from between days to months. And then within that period, the thermal camera or the thermal infrared monitor is able to capture stressful events within that time period. So it is worth exploring more and going deeper into the utility of thermal infrared imaging. Now, I wouldn't talk so much about the study side because Sami and other presenters have spoken much about the site. The method that we are using is that we are using the thermal infrared camera made by Fluke. Is a Fluke RSC 300 wide angled lens, and it has a resolution of 320 by 240 pixels, and it operates within the thermal infrared range of 8 to 14 micrometers. And this camera has been in operation since 2021, capturing images every 10 minutes, and is controlled by a software called LabVIEW. And we also are privileged to have data from the site, the BIFoR site, many thanks to Nick and all the great technicians who are always making sure that the camera is working and we are having good data. Now, this is the aerial view of the camera. So this is a camera on top of the central flag tower. And we also have a visible light camera, which also captures normal RGB images for us to compare with the thermal infrared images for analysis. And this is the view that the camera is capturing. So it captures one of the treatment arrays, which is array 6, and one of the non-infrastructure arrays, which we are using as our control. Because the camera is fixed and it doesn't move, this is the setup that we have at the site. Now, this is the view, the lab view window showing. This is the visible image captured by one of the normal visible light camera. And this is the thermal infrared image by the Fluke RSC 300. And without even delving into any analysis, it can be seen or it can be observed that the oaks within the treatment patch appears to be warmer than the ambient area, which is very interesting to look at. Now, with the workflow for the analysis, we download the data from the camera, and then we transform them in GIS and then R, and then we use regions of interest. We select regions of interest within the ambient patch and then also within the treatment patches. And then these are run in a custom code in R to extract and average the temperature pixels over the whole study period that we are looking at. And we follow steps outlined in Albert et al., who have done an extensive work using thermal infrared cameras. In addition to that, we also did some leaf tolerance leafy tolerance measurements, where we sampled leaves from both the up and then the sun and then shaded canopy of leaves. And we did this in June and July when we wanted to compare how the thermal tolerance changes with time. So we were fortunate to sample some leaves in June 2022, and also after the heat wave in July, during the leaf sampling periods at BIFoR. And we followed methods by Kraus et al. To understand how leaf tolerance changes with time and extreme events. So with the canopy temperature monitoring, we have data from 2022, from June to August. From June 2022 to the end of August. And then so the temperature, the canopy temperature throughout the season seems, or is mostly higher than the air temperature across that period of study. But we find that the elevated canopies are much more higher than in the ambient areas during most part of the day. And then also in the night, canopy temperatures are much cooler than air, and the elevated patches or the elevated areas also, just a little bit colder than in the ambient area. So this is the pattern for 2022, and the heat wave event was captured in July and also in August. And we are still working on the calibrations. I'm fortunate to be granted some funds from ECT to acquire leaf thermocouples to actually measure the leaf temperature, and it's all geared towards calibrating the measurements. Now, this is the canopy to air temperature difference that we also have. So for the canopy to air temperature difference is the ambient canopy temperature minus the air temperature, and also the elevated canopy temperature minus the air temperature. And the canopy to air temperature difference is much higher in the elevated patches than in the ambient patches across the seasons. Now, going forward with the analysis, we did a PET test to compare how canopy temperatures, or how high or what maximum canopy temperatures was reached in 2022. And then we found out that canopy temperatures in the ambient arrays reached a maximum of about 38 degrees, with a mean of 32.8. And the canopy temperature for the ambient patches was about 4.3 degrees when temperatures were above air temperatures, when air temperatures were above 4 degrees, about 25 degrees Celsius. Now, in the elevated patches, canopy temperature were up to a maximum of about 40 degrees. And then the mean for that period, or during that season, 2022 season, was 33.9 degrees, with a canopy to air temperature of about 5.4 degrees. Now, moving forward, we also realized that the canopy temperature increased with air temperature, and this was much more, seen in the elevated patches as well. We also compared the leaf tolerance measurements for June and then July, and we realized that there was, after the heat wave, there was an upregulation of leaf tolerance for both the ambient and the elevated, even though there was no significant difference between the two patches. And this is shown in this graph. So the maximum temperature tolerance increased by 2.4 degrees between June and also in the July sampling. And when we analyze the temperatures before the heat wave event in July, the mean temperature was about 15 degrees. And then after 3 weeks, the tolerance increased. And then before that second measurement, the average temperature was about 17.9. So this means that the increase in temperature, the exposure to heat waves, the heat waves that the exposure of oaks to heat wave might have, increased the tolerance, they upregulated the tolerance of the leaf of oaks. And that was seen in the subsequent measurements in the late July. So in answering the research questions, yes, when temperatures are up about 25 degrees, canopy temperature also increases. And also whether high CO2 affects the temperature range, yes, the high CO2 increase the temperature in the elevated treatment by about 1 degree Celsius. And then what was the upper temperature tolerance of Cocos River? It was ranging between 45 and 52 degrees. And whether the CO2 influenced the thermal tolerance, we did not find any difference with that. And then we also wanted to find out whether the upper temperature tolerance increased with heat wave exposure. And that is also true. It increases about 3 degrees Celsius. So implication for oak forests from this kind of experiments, the cocos robot appears to have high temperature tolerance and they can quickly acclimate from the results that we have shown. And then as the temperature also gets warmer, canopy temperatures will get warmer, and this will have some oaks will be able to tolerate to a higher level. The increase in tolerance will also have effect on also some of the physiological processes of plants, which needs to be considered in carbon balance and also in current temperature analysis. And then the maximum heat tolerance could also be included in models that consider leaf temperature measurements. Similar work is also going on, and I've also received a grant from the Royal Forest Society to undertake some research into other species to know more about other species, how other species will also tolerate heat during this season. Similar work is also going on in Ghana, where canopy temperature is also being measured. Temperatures are quite high. Here we have temperatures reaching 40 degrees at the site that we are working at. And most of the species have very high temperatures, more than the air temperature. So it will be interesting to also look at these kind of measurements. And also, we are also undertaking some trait measurements in order to parameterize or understand the mechanisms behind the temperatures that we are also observing in the tropics. Thank you.
Thank you very much, William, for the very insightful talk. We do have about 5 minutes for questions, so I would like to invite the speakers to come to the front, please.
Hello. Hello.
Okay, so first question that we have for Sami is, thanks for this excellent talk. How are you going to explore if there is less organomineral association under ECO2 than ambient CO2?
We haven't done the density fractionations to know whether the mineral associated organic matter, which is relatively more nitrogen rich, if that is the prime target for nitrogen acquisitions. And something we proposed, and probably this is our next step to look at the relative fractions of particulate organic matter and mineral associated organic matter, and to look at the changes in the stable isotope nitrogen signatures to know whether the stable nitrogen pool is being mined for the nitrogen acquisition. It's an interesting question, and this is something we want to do in future.
Thank you. We have another question for Professor Neil about isotopes. Do you think that stable isotopes may be used to distinguish between annual rings and developmental rings in 385 million year old woody plants? Here I'm thinking about ancient plants with wood, which have no living analogues. So we don't understand if the rings are caused by branches falling off or some other developmental factor.
So again, really interesting question there. I think it's from an analytical point of view, it would be possible to measure old wood, and even wood that's encased in silica as well. If the carbon is actually in there, we can get to that with the laser sampling. That would require the material to actually be preserved. In terms of trying to work out exactly where it came from in the tree, I think you might have more luck with sort of looking at the wood anatomy, if there's reaction tissues or changes around the sort of circumference of the sample that may indicate if it's grown under tension. But I think it'd be very difficult to say for sure. But in terms of analysis, if there's carbon there, it should be possible to measure it.
Great, that's good to know. We have one more question for Sami, Professor Sami. Is anybody measuring leaf level photosynthetic water use efficiency, CO2 uptake per H2O emitted?
Yes, so one of the current postdoctoral fellow who is not here unfortunately, but she's looking at stomatal conductance, water use efficiency in photosynthetic updates. It's ongoing since the start of fumigations, but it continues.
There's a result reported in
In the publications there that is published in 2022. Which includes it's a global paper and it covers BIFoR specifically in terms of stomatal conductance.
Okay.
Yes, 2022 is
Gardner, yeah.
Okay.
I could point you to the publication up to the break.
Right, we do have a few more questions, but I'm afraid we've run out of time. So I hope people that have questions will still have an opportunity to ask the speakers after the keynote speech. Thank you very much.
Hi, everyone. We're going to take 5 minutes now just to reset and prepare ourselves for the public lecture from Professors Bradwell and McIlwain. So please just a very brief comfort break, if you don't mind, and we'll be letting other people come in and join us. And then we'll try and start as promptly as possible at the top of the hour. Thank you.
Jenny first, yeah. Perfect.
No, it's showing us fallback crystals, so yeah, it's fine. All we do is just switch it off and open on the fees.
Running on. No, it's been helping. And the other thing we were getting that we weren't sure about was we're getting a kind of buzzing noise. Yeah. I think that's going to be going to.
All right. Okay. Because we're so it might be causing some sort of like virus experience.
Yeah, because at some point you had to move this marathon over to here. That's why we had interference.
Yeah. There are this avoid microphones.
All right, so charge.
Yeah. That one's fine. I'll just sit there. So this is going on now to about 5. Yeah.
So this at some point you switch it on.
Yeah.
And then this is now not muted, right?
Yep. So it's so that's mute and that's yeah.
So the unmute is actually
on the left hand side. Yeah. That's right.
Okay.
Yeah, it's not. Can't they use this? Can't they use the mouse? I think you can. Yeah. I'll use the mouse. You could put the mouse there. You've got to stand here.
Yeah, I mean, don't feel.
The mouse goes in, it goes on both screens.
Yes.
Yeah, you see it there. See it there.
Yeah, the mouse is fine. Yeah, the mouse is good.
That's it. That only goes on one screen. Yeah. We want that to turn this way.
Yeah. If you want to do foundations,
Yes, I'm studying.